you came here as a pair of scientists. Because, you know, usually in the lab are starting with one independent scientist building the group, and whereas you had a quite unusual, you know, scientific relationship, and you were doing it together. So how did you get to know Yanni and organize that way?
The idea of the leadership here was that even if the real focus here was going to be on developing molecular techniques, ensuring the development of molecular biology, that having in-house some people who were doing biology, who knew model systems, who were driven by biological questions, would be a very useful thing. And so we were both offered positions in a relatively small laboratory space but certainly enough for us to do the kind of experiments that we wanted to do. And so we both arrived on the same day. We knew each other, we were friends. We each had our own projects and our own interests, but we also knew that one of the important things for us was to use the opportunities that the EMBL provided to do the experiment that we had talked about in the past, and to actually try to push Drosophila genetics to a level and to a saturation where one could claim to know most of the parts and see how everything fit together. And so it was a great opportunity for us, and we were fortunate to be able to do that.
Usually to reach saturation, you need to repeat the same experiment again and again. So how hard it was practically to do the screen?
Yeah. So there was enough known in Drosophila that there were, interestingly, if one looked at with the available molecular techniques at the time, and looked at the heterogeneity of RNAs that were detectable, and therefore how many different genes were being produced at any time. The estimates for the number of genes was in the 100,000s. So a large number of genes, largely because we didn't fully understand genetic structure or RNA or any of those things. If, however, the geneticist, and this was an interesting conflict at the time, the geneticist and there had been certain experiments where a Drosophila geneticist would take a tiny little region of a chromosome defined by a deficiency, and then ask, this deficiency might represent 0.5% of the total genome, and ask how many mutations, how many genes could be identified in that little region. The number of genes that you identify as functions, so you knock out a gene and something happens, the number of functions was very small. And the total number of genes wasn't in the 100,000s, it wasn't 50,000, was closer to the 3,000 or 4,000 essential functions. And so we knew that. And so once you know that number, if you believe it, if you take it seriously, then, and if there are technologies that allow you with an efficiency of maybe 1 in 1,000 mutagenized chromosomes to knock out a gene, then you can calculate that, well, to discover everything in all genes in flies, you really only need a couple thousand chromosomes, a couple thousand mutants, and the experiment becomes doable. And I think that realization, and the realization that it was doable, and that it would be valuable to try it once, was the thing I think that drove us. Hey, I'm personally a scientist who may be fascinated with ideas and fascinated with deep scientific discussions, but I'm very practically driven, and the moment I see that something is doable in the lab, and I see, one could approach this fundamental question by doing this, it becomes very, very attractive to me. I will do an experiment almost simply because it's doable, because all things are interesting, and you do an experiment and you get an interest, you get a result, and you understand something you didn't understand before. So even if I look now over my kind of longish career in science and look at different kinds of things that I've done, it's often hard for me to identify a particular thread or intellectual problem or specific issue. It's been more that I've suddenly seen, if we can see this, if we can visualize something in this way, if we can make these kinds of, if there's this reagent available, we could do this fundamentally important experiment. And so I think other scientists can be more driven by some fundamental question that they recognize as central and can spend their entire career building on that single question. Whereas I think probably even more so than Yanni, I'm more driven by what is doable. One of my immediate reactions after receiving the Nobel Prize, this is 30 years ago, so this is me reconstructing, but I know this was true. I was happy with the science I was doing then. I was excited about what was going on in the lab, and I knew that my first job was to ensure that the Nobel Prize didn't change anything in my life.
Okay.
And that I could go on and change, but that I would change, that the changes would be driven not by a prize or a distant group of Swedes decided to, but that my life was going to change, as it inevitably will, but it would be changed by things that matter, by science and new scientific discoveries, by personal relationships. So I wanted to keep my life as though I weren't a Nobel laureate. I was definitely a goal in my mind. And so we put that thing into some box somewhere and haven't seen it since. Now, the reality, though, is that I am a Nobel laureate. And that probably has given me the power to have made that decision, a power to construct, to maintain a certain scientific story and a certain freedom.